Hey everyone, welcome back. This is going to be the third of the biology crash course videos. I'm hoping to make this one under an hour, but we're going to see how we're going to go. We're going to go through all of the different body systems that are relevant to the GAMSAT. I'm going to try and give a very brief, but hopefully very pointed summary of everything that you would potentially need. It's not going to be content that you need to memorize in any way, so you can probably watch this in a pretty kind of casual fashion, hopefully. You don't have to be necessarily taking detailed notes or anything like that. Realistically, most of the information would probably be presented in the STEM to you again, but the goal of this is to give you an intuitive and very quick pointed understanding of all of these points or all of these topics, so that when you get to STEMs on these particular systems, you don't really have to read too in-depth in the STEM and you can get straight to the more important stuff. So that's pretty much it. As well as that, for anyone, like obviously you guys, most of you watching this are from a non-science background, or at least for this one, a non-biology background, might also be helpful in kind of putting in some of the foundational layers as to what you might be studying if you were to go into medical school as well anyway, and take a little bit of pressure off the first year, the preclinical years. So we'll dive right into it, but there's a pretty long list. So I've made like a bit of a checklist here. We're going to go through the cardiovascular system, both cardiac cycle and the circulatory systems as well. We're also going to go through the respiratory system, the gastrointestinal system, the urinary and renal system, then also what else is there? Go through the hormonal system, the nervous system very briefly as well. So those are all the main things. There's a lot in there. So we'll see how we go. So we'll start with anatomical diagrams and anatomical terminology. These come up a lot. Again, I don't think that ASA would ever really be expecting anyone to have expert knowledge in these and to not have them defined, but again, to help with the reading, being familiar with the words is useful. So the first thing is with anatomical terms, as we have a bunch of different terms. So if I just draw out, for example, a little blob person here, I'm just going to put their arms, well actually I'll put them in the anatomical position. So here we go. It's going to test my drawing skills. We're going to have an arm and a torso. Oh, we got a bit of a bend there. Oh well. Then their feet. Like this. A very oddly shaped person, but there we go. This is the anatomical position. Technically, actually, their feet should be turning up, toes pointing straight up at us. But let's say they're just lying down on their back or standing directly in front of us. This is how they're supposed to be. And hands should be palms facing forwards. So the thumb would be out to here if I draw in some weird looking little fingers. Oh, that's very creepy, but this would be the thumb on the outside, and the same thing, the thumb would be on the outside on this one here, but I'm going to get rid of those because they're weirding me out. So first thing is we have a couple of axes that we look at, and we describe everything in relation to it. So this line here is the midline, makes a whole lot of sense in terms of its name. And then we describe things relative to where it is to the midline. We've also then got not really a true axis, but we could have like a horizontal line. And so therefore we can then talk about things either being superior, so higher than, or inferior. Now that's not to say that something below the line is not say superior. It's all relative to a position of something. So if we're talking about the knee, then we would say that the foot is inferior to the knee and the hip, for example, is superior to the knee. So it's really just a positioning. You could imagine that you could think of this as really sliding depending on the position of the object that we're trying to name everything in relation to, like that. I'll get rid of that one for the moment though. The words will come back in a moment. And then as well as that, we have some other ones. So we've got like, you've got like cross sections, which is if you were to, it's quite gross, but if you were to like chop the person directly through their middle, through their torso or something, as we were drawing there. And then you've also got the coronal plane as well, which is like going right down, right through the middle of the head, facing right through. So kind of separating the front from the back. So if you're from a non-science background, this might be a little bit uncomfortable, a little bit gross to think about, but it'll all be something to get desensitized to anyway. So from there then we've also got, so we've got superior and inferior, meaning higher and lower, fancy ways to say that. We can maybe make an axis like this. And then we've also got, we've got left and right, that doesn't change. But if we're closer to the midline, then we call it medial. And if we're closer to the outside, then we call it lateral. So medial and lateral. So, let me get the spelling right. And so, for example, the hand is lateral to the torso, right? Because it's further out to the side. Likewise, if you've got your hands facing out, I'll just do it like this, then, no, that actually doesn't work. I'll just do it like that. So if the anatomical position, the midline's here, so then the thumb is distal, or sorry, the thumb is lateral to the midline, whereas the pinky, for example, is medial to the midline. It's closer to the midline. Then we've also got the concepts of anterior and posterior. So this is referring to effectively front and back. So we're looking at the anterior position, but you can imagine that on the back side, that would be the posterior position, like that. So we've got all three dimensions with words that we can use. So up and down, superior, inferior. We'll just say the horizontal plane, medial and lateral, because keep in mind over on the far left, this would also be considered lateral, and medial would be closer to the midline again. And then we've got front and back, posterior and inferior, posterior and superior, posterior and anterior. There we go. There's also some kind of old school systems that are used in animal anatomy as well. And sometimes they're used in some of the ACER questions, so look out for them. But again, realistically, they'd probably explain them. So sometimes we refer to the concepts of dorsal versus ventral. And really that's just an alternative to saying superior and to saying anterior and posterior. So ventral is like anterior in the anatomical position, and dorsal is like posterior. And so you can think about, for example, in like fish, they refer to like the dorsal fin, and that would be if you've got a fish like this, there, and then here's its mouth, and here's its little eyes, then it's got a dorsal fin like that, because it's on its backbone. So coming from the dorsum, or the backbone there. So the same thing, our spine is on our dorsal side, and then our stomach, I guess, in our front, it is on our ventral side, or our anterior side. Another way to look at it as well, the anatomical position is like that, so that everything curls inwards on the anterior or the ventral side. So you can also think that if I was to make like a C shape with my hand, the inside of the curve must be the ventral or the anterior side, and then the outside of the curve is then going to be the dorsal or the posterior side. And you can check that with everything. You can do it with like the elbow joints, you can do it with the back, all those kind of things. The outside of the curve is posterior, inside is anterior. Cool. And then the other one we have is proximal and distal. And again, we use this in place of medial and lateral, but it's also not necessarily related to the midline. So the idea is that proximal just means closer to, distal means distant to. So if we're talking about, for example, the elbow joint, then the idea is that the forearm is more proximal to the elbow than the fingers are. The fingers are distal to the elbow, they're further away. That's it. Cool. So those are all of the terminologies that we use. And then we've also got movement. So a couple of different movements that we need to be mindful of. So we've got flexion and extension. This one you might already be familiar with. So flexion is the curling inwards, extension is the straightening out. So you could do it with your fingers, you can flex the finger, and then you can extend the finger. You can do it at the elbow, you can do it at the knee. The weird thing is that's the one that probably breaks the rule, is the knee joint does go backwards. So I mentioned before about the bending of joints. Most flexions curl forwards if we're in the anatomical position. However, at the knee, flexion of the knee actually bends the lower leg backwards in that case. Again, realistically, you're not going to be tested on the use of these terms. It's just being familiar with what the words mean, because they may use them in a STEM. There we go. So flexion, in other words, is decreasing the angle, and then extension is increasing the angle. So if we just think of a joint like this, if we swing it inwards, that would be flexion, because we'd be reducing the angle in here, and then we can extend it back outwards. That would be extension, like that. Then we've also got abduction and adduction. So that would be, for example, if you were to swing your arms out to the side, then you've got abduction, like this, because you're subtracting it from the midline, and then swinging it back towards the midline, that would be adduction. So you're adding it back, is a simple way to think about it. And you can do that with the legs or with the fingers, you could splay them out. And then you've also got pronation and supination. So this is to do with a twisting action. So pronation is an inward twisting towards the midline. And then supination is obviously outward. So a really simple example is you can do it with your forearms. If you've got your arm in the anatomical position, so palms facing forwards, if you spin your forearm over at the elbow joint, I guess, or through the forearm, the thumb is switching over and facing now to the median or the medial side. That would be pronation, so rotating inwards, flipping it back out again, that is supination. So pronation inwards, supination outwards. There we go. Cool. So that's all of our anatomical terminology and physiological terminology. So now we can get into the different systems. So first of all, we're going to look at the cardiovascular system. And so it's kind of got the two parts in the name. You've got the cardiac system, the heart, and the vascular system, the veins and the arteries. Put that together, you've got the cardiovascular system. Again, we don't need to know a huge amount of technical knowledge. We just need to get familiar with the anatomy of it and the basic function of each one and what to be looking out for. So first of all, let's start with the heart, and we'll start with the anatomy of it. So I'm going to do a simplified diagram of a circle representing the heart. And then what we can do is we can just chop the heart into 4 quarters. Realistically, the heart has a somewhat kind of triangular, conical kind of shape, and it kind of sits at an angle, which makes it kind of annoying to draw, and it makes the explanation a little bit messier. So we're just going to simplify it like this. Now, the first thing is in an anatomical diagram, because the person, if the heart was literally in their chest, they would be facing us. So left and right are inverted. So what you'll see is on the left side, you'll actually see the right labels of things, and the left side will appear to be on the right side. So it's all inverted because we're not facing the back of them, they're facing us. And we split it into 4 chambers. So we have the right atrium, and then we have the left atrium. So these are the chambers at the top of the heart, and then below we have ventricles. So again, we have a right ventricle, and then we have a left ventricle. And you may already be familiar with some of these words. So then the next thing is we need to allow blood to be able to transport between them, but not between everything. So we have to have valves that open and close. So we have a valve between the right atrium and the right ventricle, and we have another valve between the left atrium and the left ventricle, like this. And so from here to here, we have the tricuspid valve, and then over here we have the mitral valve, sometimes called the bicuspid valve. Where that comes from is actually down to the way they're structured. You don't need to know a huge amount about this or even anything about it. They have little spindly fibers that are attaching to the muscle that's going to pull and draw them open like a drawbridge. And the tricuspid valve has 3 of those. So it kind of ends up looking like this, and each of those little strings are attaching downwards and pulling downwards and opening up the trapdoor. The mitral valve has just 2, so the bicuspid, and the idea is that it often gets the name mitral because it looks like the mitre, which is the shape of the crown from a king or a queen's crown. And so again, from those 2 spindly fibers pulling it open. There we go. Cool. All right, I'll get rid of that detail though. So we've got the 2 valves there. Then you've also got valves opening or openings into the atria and out of the ventricles. And what you'll find is the blood flows from the atria down to the ventricles, back out, back in to the atria again. So this is the general kind of structure of the heart that we're dealing with. Before we get into all the circulation, we've also got as well to consider the walls of each of these chambers. So you'll find that the atria at the top have very thin walls, not a whole lot of musculature in them. And so they're under relatively lower pressure than the ventricles are. The ventricles are a little bit thicker, their walls, they have a lot more muscle on them, and so they can contract with a much, much greater force and therefore increase pressure a whole lot more. And the left ventricle in particular is particularly thick in its wall, and so therefore can produce a lot more pressure. We'll come back to that, but that's something to be mindful of. So now when we get into where the blood is actually going, so it always enters into the atria like this. And on the right side, we're going to be receiving blood that is from the body that has no oxygen left in it. And so this is from the vena cava, like that. And we don't really fuss about the names of the openings here. So the vena cava is just a gigantic vein that drains all the blood from the body into the right atrium. It's technically made up of two parts. It has two connections to it, but we don't need that for the diagram. You've got the superior and the inferior. So the superior is above, draining everything from above the heart, and then the inferior is doing the bulk of the work, draining all the blood coming back up from the rest of the body below the heart line, like that. We can think of them as just merging and joining together, and they filter all their blood straight into the right atrium to return it to then be recycled and used. So this is our vein system. We'll just put V over here. Then on the left side, we have blood entering from the lungs. This is the pulmonary vein. Over here, we then have the pulmonary artery exiting, and that will then carry it to the lungs. From the lungs, and so from the left ventricle, blood will be exiting, and this will be the aorta, like that. So we have all these ins and outs. So now let's connect the pieces, right? So the pulmonary artery will carry blood to the lungs, and let me just draw in, I'll do my best to do a nice little diagram of two little lungs here. There we go. They will pick up oxygen and then carry blood back. So the idea is I might actually do this in red. Red is usually used for oxygenated blood, and blue is usually used for deoxygenated blood. So out of the right side, you have no oxygen on the right side of the heart. It's all been spent. So you got no oxygen. It goes to the lungs, it picks up oxygen, and now we'll go to red, carrying oxygenated blood towards the heart. That can then go through, out like this, out the aorta, and that will then go to the body, and it'll be spent. We'll use up the oxygen. So that'll be mostly used at the muscles and at the other tissues. So just draw like a little striated muscle belly like that, and that will then return back there. And so you can imagine that this will be oxygenated out of the aorta. And then once it goes to the tissues, it's going to spend the oxygen. It'll be deoxygenated, which explains why it's deoxygenated when it comes back in the right side. And so this is our full now circulation going on. And so over on the right side here, this would be referred to as the pulmonary circulation, to do with the oxygenation of the blood. And then over on the left side, this would be our systemic circulation. If ever you see those words, that's what it's referring to. That's the spending of the oxygen. So there's a few little patterns that we can pick up here as well. So we'll keep those on there. We can see, first of all, only oxygenated blood on the left side and only deoxygenated blood on the right side, right? Technically, there may be some oxygen still returning back that never got spent and all the rest of it, but generally speaking, this is how we should see it. So that's one thing that you can take from this. The second thing is, you'll notice the ventricles are allowing blood to exit and the atria are collecting blood. That's another thing we can do. You'll also notice maybe a pattern in the use of the words artery and vein. Arteries are always carrying blood away from the heart, and veins are always carrying blood towards the heart. So you can see veins drain into the top, into the atria. Arteries, which aorta is the main artery as well, the aortic artery or the aorta, these are exiting, allowing blood to leave, right? Arteries and veins are not named based on oxygenation. That's a very common misunderstanding. People think that arteries means oxygenated, but it doesn't. You can see that, for example, the pulmonary artery carries deoxygenated blood, like that. Cool. All right, so now if I get rid of some of that scribbling, because it's a little bit messy. All right. So the next thing is just looking at the functionality of where the blood goes. You've actually got both the left and the right hand sides working simultaneously. It's not that one's working at a time. There's two phases of the heart cycle. So you've got diastole and then you've got systole. Diastole is relaxation, systole is contraction. And it's all happening kind of at the same time. It's coordinated, but things are happening in a particular sequence. So we'll start with what's happening when the blood is filling. when the blood is filling into the atria, it's generally under very low pressure and it's just kind of dropping in like this from the two veins draining in. And that would be when the atria are relaxing. They're in atrial diastole, right? So they're relaxing, allowing the blood to fill in. During that time as well, you've also got down the lower half, you've actually got systole. You've got contraction at that point. And you've got the walls contracting inwards on the ventricles to try and tighten them up and squeeze and push the blood out of the arteries to each respective area. So you've actually got contraction and relaxation happening simultaneously in two halves of the heart, the top and the bottom. And so then from there, then you'll have the ventricular diastole, and that will be where they relax. And instead now it'll be the atria's turn to contract. They will be under systole. So it's coordinating one then the other, right? Squeezing and then not squeezing. And so when you've got contraction at the top, whilst the ventricles are in diastole relaxing, that'll allow the blood then to push through and fill the ventricles like that. And so when that happens, then they fill up, the pressure goes up, and then they squeeze, and then back into the second cycle, they then put the blood back out again. Cool. All right. Cool. And that's everything for the cardiac cycle. In terms of circulation, we've talked about most of it here. The only extra thing to know is that with the blood vessel hierarchy, we have a bit of a breakdown. So I'll move this out of the way. We start with arteries, which are the largest, and they've got pretty thick muscle walls. They can contract pretty well. Then after that, you've got arterioles. And what is happening is the arteries branch, just like a tree branch, into smaller but still relatively large chunks. These are your arterioles. The blood then diverts like this. And then arterioles can go further into capillaries, right? You can technically break them down even further, but then they start to separate into very, very small little vessels like this, and these are your capillaries. And this is where we're going to get gas exchange at the tissues. On the other side though, on the receiving end, you've got capillaries joining in, and this is then forming a capillary bed. I'll come back to that in a moment. And then the blood will actually start to go back out into slightly larger little mini veins. So these are your venules. And then from there, venules can join and then form larger veins. And this then leads back to that superior and inferior vena cava, which are the primary, very, very large veins like that. And so this might be going off and forming its own network with some other ones and then forming another vein like that. So the blood is making its way through. What is happening in the capillary beds is this is where we've got gas exchange. So on the left side here, we have oxygenated blood. So we've got oxygen carrying through. And then at the tissues, at the capillary beds, we then have oxygen being used in metabolic processes. That's why we need to breathe in oxygen. And it's converting oxygen into carbon dioxide. And the carbon dioxide is then going to be carried through the blood, now deoxygenated back to the heart, and then it'll be recycled at the lungs. So then once it gets to the lungs, we've got the reverse happening. Then we've got the veins carrying, technically it'll go via the heart. So I'll just put heart like this in the middle. And then out of the heart, we have the pulmonary circulation. It then goes to the lungs, which I'll draw in my nice little diagram again. There we go. And at this point, this is where then we have that carbon dioxide being exchanged. So we'll do it in blue, keep the color coding. Carbon dioxide comes out, we breathe it out, and then what do? We breathe in oxygen gas, and then that's going to go back in, all the way back around, and then restart the cycle. You've got oxygenated blood again, and it's just going to keep going. So that's the concept of then gas exchange. So you can imagine it's going to go back to the heart, and the heart then is going to, I keep misspelling that, the heart then is going to pump that blood back into the arteries, and then you've got a full cycle. So it enters the heart and exits twice in a cycle overall. Cool. There we go. So that's the cardiovascular system. So then next up is the respiratory system. So we're really not going to look at a huge amount here, just a brief amount of the anatomy of it. So obviously the upper and lower airways, people often think about the lungs as still being upper because they're higher up in the body, but they are lower down to the whole airway system. So the lungs are actually in the lower region. You've got the mouth and the nose as the kind of upper airways, and the trachea or the windpipe, that kind of thing. So if we've got mouth and nose like this, I'll put some little teeth in there. Oh God, this is creepy. I shouldn't do diagrams, but anyway, we've got air coming in here, air coming in here. They can join up, go down the trachea, or the trachea, however you want to say it. I always say things weirdly. I think I was taught by people at uni that said things weirdly. I always get made fun of by my VCE students. And then it'll go down and it'll go into the lungs, which will separate like this. And then you've got the rest of the gas exchange. So what we really want to know though is just a little bit of the anatomy. So we've got the main trachea here, then you've got the left and right bronchi. So bronchus individually, bronchus here, and these are just big trunks. And then they actually branch into bronchioles, just like the arteries and veins did. And in fact, they have 27 layers. If I remember right from my undergrad, I remember being taught there's 27 layers of that hierarchy before it gets all the way down to these tiny, tiny little balloon structures that bubble like this, like bunches of grapes. And these are called alveoli. So each one of those is an alveoli. And all we're doing with that in that system of the anatomy of the lungs is increasing the surface area. So we know that in everything in bio, if you increase the surface area, then you increase the reactive surface, which means you get more reaction. And in this case, we get more gas exchange. And a really interesting fact is that the total surface area of the lungs, of all of those alveoli, if you were to kind of splay them all out open flat, is 30 square meters, which is about the size of a tennis court, all inside just a pair of lungs on the average human. Then if we go a little bit more into what is actually happening, so in terms of breathing, the mechanics of it, there's a few things that are happening. So you've got the diaphragm, so a muscular wall that is pulling downwards and creating negative pressure and then drawing air inwards. Same idea as like if you've got like a rubber glove on your hand, you pull up like that, you can feel the suction on the hand. It's the same concept. So it's trying to suck inwards. It's trying to create negative pressure so that air rushes into the lungs and fills them. So that's happening underneath. You've got a diaphragm here. It pulls downwards and then it tries to create negative pressure and to then draw in the air. The second one then is you've also got breathing muscles as well. So you've got things like intercostal muscles. Again, you don't need to memorize any of this. This is just so you're familiar with the words. This means just between the ribs. And they're all working when you're breathing to control the size of the rib cage to then increase and decrease the pressures and also allow for greater expansion of the lungs to kind of blow up. And the other thing as well is linked to that is you've also got some other postural muscles that are involved with it as well and around the chest and around the back, which help pull the ribs open. And the idea is that there's an analogy of the bucket handle. If you've got a handle on a bucket like this, representing the rib cage, if you want to swing the handle up, what does it do? It goes out and upwards like this. And this is exactly what the ribs do when you're breathing in, is they bend up and outwards like that, winging almost, to increase the volume of the rib cage. And you can think about it like if you've ever like gone for a run or something like that, or you're out of breath, the first thing that you do is you either put your hands on your hips like this to try and hold it up and support it and then raise the ribs, or you might be outstretched and have your hands out on a wall or on a fence or something, and then you usually drop your head and kind of lean downwards, and what you're doing is trying to arch up and pull up the rib cage to increase the volume and maximize the inflation of the lungs to get as much oxygen in as possible. All right, so there's also something else in this which is called spirometry, which is to do with the measurement of the volumes of the lungs. And this is realistically the thing that they use the most. They will explain all of it in most cases. They won't expect you to come in with a complete theoretical understanding of it, but it's good to go in with an already existing familiarity with it. So if we were to draw a graph like this, and in fact what I'll do, we'll draw a graph like this, that's measuring the volume of air in the lungs. Normally what we have is a particular volume that we kind of fluctuate in. And so this volume here from here to here is called a tidal volume. And this is just your standard sitting still, normal, regular daily breathing, right? From that though, we know that if we need to, we can breathe in quite a bit more. We can inspire quite a bit more. So we could also spike it like this and then come back down. And this excess volume from here to here, this would be called an inspiratory reserve. So it's the extra volume that we have on reserve if we need it to breathe in even more. So a simple way to do it is if you just breathe in normally and then just hold your breath and then try to suck in more air, that extra volume is your inspiratory reserve. Like that. And it's often just called IRV. Let's do that in tidal volume TV. Same thing, if you breathe out normally and then hold your breath before you breathe in, you can actually find that you can keep going and you can kind of push out even more air. And so there, that excess is going to be your expiratory reserve volume, but you won't be able to completely empty the lungs when you expire. And so that excess little bit there is usually called a residual volume, and that is the little bit of volume that you can never push out of the lungs. And this is because air will always occupy a space in that way, right? And that is just, generally speaking, a measure of the air, that the basic volume of the anatomy of your lungs and airways, that kind of thing. And then what you can do is combine all of this. So if we went all the way from lowest exhale to highest inhale, this would be what's called a vital capacity. So if you're absolutely needing your lungs because you're going to die and it's vital that you use all of your volume available, that is your vital capacity. And then if you were to take the vital capacity and add in that residual volume that we can't really access, that would be our total lung capacity, TLC. There we go. Done, like that. And that's our spirometry. So you might get given a graph like this, but being familiar with it immediately really massively saves time. And you can see you can create little formulas. So you can say things like vital capacity equals tidal volume plus inspiratory reserve plus expiratory reserve volume. You could say that total lung capacity equals vital capacity plus residual volume and that kind of thing. And then you could like mix and match and blah, blah. Cool. All right, moving on from there though, that's it for the respiratory system. I'll try to keep that pretty short. So then we have the gastrointestinal system. Draw that in. And so with this one, let's again start with a brief little tour of the anatomy of it. So we start technically at the mouth, then we've got the esophagus. If I can spell that right, I think I've got it right there. Esophagus, mouth, and then you've got the stomach. I'm going to do my best. It's kind of a J shape. That's not drawn very well. Let me try that again. It's kind of like a bean, and this is the stomach. Out of the stomach, then you have the first turn of the small intestine called the duodenum. Some people say duodenum. I was taught duodenum, so I'm going to stick with that. Then you have the small intestines, which is usually just drawn as a squiggle. It does have a more specific shape, but let's just do this. That's a pretty messy drawing, but oh well. This is going to be the small intestine. We'll keep it going. It actually bends all the way over here. The small intestine keeps going. Comes all the way over to the right side, which looks like the left because we're looking front on. And then it meets up with the large intestine or the colon, and this is a lot bigger and it's a lot less convoluted. And you've also got the little appendix kind of hanging off there, which we don't really talk about. And then you've got the ascending colon. Then you've got the transverse colon running across. Don't worry too much about the messy writing because you don't really need these names, but it's just for familiarity. Then you've got the descending colon, and then you've got sigmoid, which kind of runs inwards, S-shaped a little bit, and then out, and then you'd have rectum at that point at the very end. So sigmoid colon, like this. And along the way, we also have some accessory organs. So you've also got the liver, which sits kind of over like this, kind of like a big triangle like this. It's probably way out of whack in terms of size. And then underneath it, you've got a little balloon called the gallbladder. like this. This is the liver. And they're going to interact and actually help contribute to what's going on in the duodenum. We'll come back to the functionality there. But that's the basics of it. So it's really just one big long tube, right, overall. And it's kind of weird as well, like if you think about it, the body is just like one big tunnel, basically all the way through. It's just one big tube, which is, yeah, very, very strange. The other thing is, technically, I always think about this, the inside of the intestinal system is actually the outside. So go figure. But anyway, so if we were to go into then digestion, so there's 4 different elements of digestion and metabolism. So we've got mechanical, we've got enzymatic, using enzymes. Then the third one is chemical, using acids and things. And the fourth one is bacterial, where we use bacteria, good bacteria in the gut to actually break things down. So if we start with the mouth, you've got mechanical and enzymatic. So mechanical is chewing or mastication, and then the other one is enzymatic. So you've got enzymes in saliva that break down complex sugars. From there, then you've got into the stomach. When you're at the stomach, then you've got mechanical, and then you've got chemical and enzymatic. So mechanical is where you get something called peristalsis. And what this is a coordinated constriction of the muscles in the wall of the organ, and it happens in the intestines as well. So first, what it does when the food meets the stomach is it just does a churning action. So it's kind of squashing and pinching like this at different spots and just kind of sloshing the stuff around the stomach. And that's just to help mechanically break it down. Then later on, when it wants to exit or empty the stomach, it'll then start to squeeze it like a tube of toothpaste, like that, and squeeze it out the other side. But before it does that, it'll also have enzymes in there like pepsin, which breaks down protein. Again, don't memorize this stuff. I'm just mentioning it so that you're aware. And then the other one is chemical. So you've also got stomach acid, which is effectively hydrochloric acid with a pH of around about 1. And that will help chemically break down food particles as well. So then once it does that, it'll then exit. It'll exit into the duodenum. And this is where our accessory organs come in. So the liver produces bile. And bile is used for fat digestion. So it's got lots of different chemicals in it, including bile salts and electrolytes. And what they do is help digest fat, because our body's not very good at digesting fat and breaking it all down. It comes in big lumps, and it's going to avoid anything that has water in it. And all of our cells are very water heavy. So we use bile, which includes bile salts, to break that down. In order to do that, though, we need to concentrate it. So the bile actually gets sent to the gallbladder first, and it sits in there and then really, really heavily concentrates. It's kind of like a fine wine kind of thing, maturing over time. So it builds up there. You get a small volume of it. It gets very, very concentrated, and then that gets ejected into the duodenum when the contents of the stomach is emptying. It coordinates, squirts that in, covers it, and then you've got a huge amount of enzymes in there and chemicals as well. And then you've got enzymatic and chemical breakdown. Then in the duodenum and the small intestine, you then have peristalsis again, squeezing it through, and you get nutrient absorption, mostly proteins and fats and that kind of thing, all of our kind of macronutrients. Along the way, if we were to zoom in on what's happening here, if this is the intestine, the inside, not to scale, has these little finger-like structures on the inside, like this, and these are called microvilli. And what they're doing is just massively increasing, they're on both sides, massively increasing the surface area of the inside of the tube, and therefore increasing access to the food particles for nutrient absorption into the bloodstream. So you're getting nutrients up and into the bloodstream just outside. That is absorption of nutrients. And then finally, when it meets the large intestine, you still have peristalsis, but you don't really have much nutrient absorption at this point. Most of the large intestine is to do with breakdown of fiber. So fiber is broken down via our good gut bacteria in the large intestine, and a lot of it is also for just water balance, to make sure that we're not getting dehydrated, because a lot of water is lost to the intestines in the process. So we're usually gaining the water back from the colon back into the tissues as well, and back into the bloodstream, I should say. Cool. And that is the basics of the gastrointestinal system. So then we have the urinary and renal system. So again, a bit of the anatomy of this first. So you've got lower down, technically, kind of like where the ribs stop at the back, you've got your kidneys, and you've got them kind of looking like this, looking like a kidney bean. That's a bit exaggerated. Like this. It's going to turn it into a circle. Let's try that again. There we go. There's one. There's two. On the top, you've got the adrenal glands, adrenal, so adding to the renals. We don't really talk much about those in this system. They're more for the endocrine system. And then out of those, you then have ureters, and ureters are little tubes that carry urine to the bladder, which is just our storage system. And then from there, the rest of the excretory system, we're just going to say the urethra, which is just the common tubule where urine will pass through and then exit the body. So here we've got our kidneys, and we're more interested in what's going on inside the tissue of the kidneys there. But this is the basic passing of urine, where it comes from. We've got blood flowing straight into each of these, and we're actually filtering the blood. So not only do the kidneys make urine, they also filter out toxins and things like that from the blood at the same time. So if we were to look at the structure of it, what's actually happening there is the kidneys are made up of little particles or little structures called nephrons. And so a nephron is made up of glomerulus, which I'm just going to do kind of like this. And it's really just a giant, or not really giant, it's really just a little capillary bed, or a glomerulus, like that, for the most part. And this is where we again get exchange of things. And so what's going to happen is you've then got something called Bowman's capsule, which kind of wraps around it like this on the outside. This is Bowman's capsule. And this is going to collect any filtrate, anything that comes out. So we're going to filter the blood, any volume that is useless that gets filtered out, our filtrate will go into here. This will be our filtrate like that. And this will become the urine volume. And that will then head down to a tube that kind of looks like this. It goes up, and it goes like that, and then back like this. And so we have a few different structures here. In fact, that's actually not quite right. Let me redraw that again. And it joins into something called a collecting duct. There we go. So we have this tubule, this renal tubule, and the more important things are this here. So we have the what's called the loop of Henle, named after the person who discovered it and described it. And so here we have nutrient reabsorption. So we're usually balancing electrolytes, so things like our sodium, our glucose, our potassium, our calcium, and all of these things. So the kidney is actually performing a really, really important function in terms of electrolyte balance and also hydration level. We're also balancing water over here. So then as the filtrate goes, it then joins the collecting duct or collecting tubule. And then what's happening here is we're maintaining our water balance. And keep in mind that in the tubule actually means exiting, because whatever stays in the tubule is going to become urine volume, which will exit the body. So when we say water going into the tubule, that is actually dehydration. Water going out of the tubule is retaining it, getting it back from the urine and putting it back in the bloodstream like that. So we have water being balanced like that. And that's really the only detail that we need to understand the urinary and renal system too. Anything else, and they're going to explain it. Realistically, they'd probably explain this as well. So again, just for familiarity. Moving on from there, so then we have the endocrine system. Now we start getting into how cells signal and communicate with each other. So the endocrine system is also a fancy way to say the hormonal system. And what we're really doing is we're creating signaling molecules that can go from one cell to another in even a distant part of the body via the bloodstream. And so we have an endocrine tissue. So this might be something like, for example, the hypothalamus, which is a small part of the in the brain, and it'll then secrete. It'll secrete a hormone, like for example, corticotropin releasing hormone. I think that comes from the hypothalamus. I think I'm right on that one. And then that will go into the bloodstream. So via the blood, that will then go to the next spot. Now, it might just go very, very close by. It might go to the anterior pituitary gland, but then that can also release another hormone. And I can't for the life of me remember what that one is, but I'll just say hormone A, doesn't really matter. And then that could even go via the blood all the way to the adrenal glands, for example. Like that, which I think is to do with cortisol, if my memory is right. Corticotropin releasing, yeah, because that's a corticosteroid. So cortisol then gets released. You don't know any of these hormone names. These are just examples. The idea is that if you want to create a message and then send it to another part of the body to act, you may use a hormonal system like that. So this is used in things like glucose regulation with insulin and glucagon from the pancreas. We've got a bunch of different endocrine tissues in the body, so tissues that can produce hormones. So we have some of the main ones that you'll come across are the hypothalamus in the brain, the anterior pituitary, just sitting below it. Then you've also got things like organs such as the testes and the ovaries for reproductive hormones and sex-determining hormones. Then you've also got, what else is there? Then you've got the adrenals, of course. So the adrenal glands, but you've also got things like the pancreas and the thyroid in the neck and that kind of thing as well. These are all tissues that produce hormones. Again, that's all we really need to know about them. Anything else would be explained in the STEM. Then the other method that we've got for signaling is the nervous system, which you'd probably already be somewhat familiar with. So we just want to kind of understand how that functions. So first of all, there's a hierarchy. So you've got the central nervous system and the peripheral nervous systems. And the central nervous system is made up of the brain and spinal cord. So it's not actually made up of nerves at all, but instead just dense tissue of neurons, the cells that make it up. The peripheral nervous system is made up of the somatic and the autonomic. And the autonomic is then made up of the sympathetic and the parasympathetic. And again, do you need to know this hierarchy for the GAMSAT? Absolutely not. But it's possible that they may call on this hierarchy, give you the hierarchy, and expect you to understand it. So this will speed that up because it comes up in first year biology. Somatic is to do with voluntary control or conscious control. So if you move an arm or something like that, you thought about doing it, it means the somatic nervous system was responsible. Autonomic is unconscious, it is involuntary. So these are things like digestion and that kind of stuff. So sympathetic is the preparation for action, and then parasympathetic is the reverse, that is the calming down after action. So a common one is parasympathetic is often called rest and digest. It's a good trick because the only thing that goes up with parasympathetic activation is digestion. If you're being chased by a bear or something, your body is not really concerned with putting energy into digesting lunch. It's focused on getting all of the blood and all the oxygen to the muscles, increasing or dilating the pupils so that you can see where you're going to run or where the bear is, and that kind of thing. But then once you've gotten away from the bear, assuming that you can run really, really fast, then all of a sudden it'll be like, all right, now I need to lower my heart rate again. I need to contract the pupils, and I need to start upping digestion again to recover from all of the energy expenditure from running from the bear. That's it. But we don't think about any of this. The body kind of goes into autopilot, so it's autonomic. Then if we look at the actual structure of the cells that make up the nervous system, so these are the neurons, and now I can finally put my neuroscience to some extent of a degree because I wasted $30,000 in debt on it and never used it. So we can draw roughly kind of star shape. This is going to be the cell body. They have these weird kind of spindly fibers, and these, I probably labeled that really poorly, these little branchings, these are dendrites, and these are trying to receive messages from the next neuron. They then branch out into this big long fiber here. This is the axon, and then this branches out further like this, and these are the axon terminals, like this. In terms of how the message actually gets across, it's more about the chemistry of it. So if we start, for example, right here, we're going to look at what happens when the membrane changes its potential. So if we zoom in, the idea is that on the inside, we have a relatively negative charge, and on the outside, a relatively positive. Technically, both are actually negative, but the outside is less negative, so it's relatively positive. And so what's going to happen is you've got really, really high sodium concentration outside the cell. Sodium wants to flood inwards, like this. The only way to really allow it to flood inwards is to open the floodgates, right? And so the way that that is done is it's done through excitatory neurotransmitters. So a neurotransmitter is another signaling molecule. It's not a hormone, but it works in a very similar way. If enough neurotransmitter is received on the surface of the dendrites, then it'll trigger the release or the opening of the floodgates and allow the sodium to flood in. When the sodium, which is positive, floods inwards, it'll flip the charge to be relatively positive inside, relatively negative outside. And then you will have a chain reaction of attraction like this, and you'll see it flip, 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 and so on, like this. And this is referred to as the action potential. And then as that depolarization, that flipping of the charge keeps making its way along, it'll eventually bounce its way along, reach the end of the axon terminal. And as it does that, then what you'll get is calcium will also flood in as a result. When calcium floods in, you'll have, in the end of the terminal, you'll have little vesicles of neurotransmitter, potentially the exact same one as what was received. And when the calcium floods in, that will trigger the release of this out. That neurotransmitter will release, and then if you had another neuron sitting beside it, those neurotransmitters get released, received, and it'll start the process again, like that. The idea is this is the basics of neural transmission. I'm not going into great amounts of detail because I don't think it's all that relevant, just to appreciate that there is an electrical component when you're inside the cell, and there is a chemical component between the cells. That's the main thing. And it's to do with the reception of a neurotransmitter, like that. So therefore, if there's anything to do with failures of reception, then therefore the message doesn't get across. If you've got failures of transmission, then it means the message doesn't get across. If you have neural damage and the nerve is cut or the neuron is cut, then you can't get the message across. These are the basics of it. There we go. So that's everything that I wanted to go through with this bio crash course and all the different body systems. Hopefully that's been really helpful in clearing up a lot of what you need to know. And keep in mind, like I always say, it's a reasoning-based exam. Most of the bio questions you don't need you don't need a lot of background information compared to, say, chemistry or even a little bit with physics. But the idea behind this crash course is to give you the familiarity with all of the concepts if you're from a non-science background or have never done bio before. So take it only as being familiar with the concepts and the basic functions of each of the systems, maybe some of the words along the way, just so that you're not thrown by it. But realistically, they will give you the relevant information in the question. The one that I would probably say you do want to know is the cardiac cycle. That one appears to be something that they assess a little bit more directly. Everything else, though, is a little bit more technical, so they'll probably explain it, but this will help speeding up the processing. Anyway, if you like this, then like the video and leave me a comment. Let me know your feedback on it, and then I'll have a fourth one coming. I don't actually really know what the fourth one is going to be yet. There's a few little loose ends that we need to tie up, like gene expression and that kind of thing, but maybe some stuff on metabolism and biochemistry as well, so we can kind of bridge the crash courses together. I'll work on that one and hopefully have that out early next week. And then we might be near to the end of all of the crash course content so that you can get started into the questions. All right, anyway, I'll leave that there. I have the joy of editing this video now. This is probably one of the longest ones that I've had to edit really fully, so that'll be fun. I will see you guys in the next video.